Okay, very good. It's 100 people, so I think it's time to start. So welcome everyone to the Cambridge Cheminformatics meeting. So I'm Andreas Bender and I will guide you through the program today. So we'll do a brief introduction first, 5 minutes or 3 minutes about those meetings. Afterwards, I will hand over to the speakers. So what is the aim of those meetings? The aim is to bring cheminformaticians, but in the widest sense together, but that includes molecular modeling as well and machine learning and bioinformatics. It's partially a historical name, so anything that relates to modeling, mostly chemical data in the drug discovery context, for example. And there's both a scientific as well as a personal component. And because we started with physical meetings in Cambridge nearly 10 years ago now, it was usually 40 people or so who turned up, or sometimes 30, sometimes 50. And we went to the pub afterwards in order to also meet personally. That didn't work anymore due to COVID, but there was one advantage because it's now much easier to participate in those meetings worldwide. So we have now on average 300 or so registrations from all over the world. And again, this time as well, I just checked that from Brazil to Japan. So I'm quite impressed actually because it's quite late in Japan. So welcome in particular to those from Japan who are participating here. And so that is the aim of those meetings. We have a few more meetings this year. So if you have time, you're very welcome to join us then as well. Please mark your calendar. The next one's on the 1st of September, and afterwards the 24th of November at the same time, 4 o'clock UK time. And if you would like to present, please let me know. You're very welcome to share your work as well, even if it's work in progress. So all the dates and registration details are on this website here, c-inf.net. And you can also register for the Cambridge Cheminformatics newsletter. So circulate that maybe once per month or so. So that's about new jobs, positions in the area, events such as Cheminformatics conferences and machine learning items. And that's on this website, drugdiscovery.net. And you can also follow me on Twitter if you wish to do that. And so the main meeting will be recorded as agreed with the speakers. And there's a particular point because we are making the presentations available afterwards in the Cheminformatics YouTube channel. So you find the link on this website as well, c-inf.net. And so usually the talks afterwards get a few hundred views as well. And in particular for people from other time zones or those who cannot make it, that is very convenient. As for questions, please type your questions in the chat window and feel free to do that also along the presentations already. Afterwards, we will go through them orally as time allows. And ideally, if we cannot answer all questions orally, speakers would ideally follow up by chat for the remaining questions. And you can also just type your name, for example, and then we will go through that in order as well. And you can ask your question orally then. Yeah, and very important, so there's a social component, although the meetings are online right now. So put your beer or other drink into the fridge because afterwards, so we will have 3 presentations now. That should be about an hour and a half or so for those presentations. Afterwards is Cheminformatics Pod Night. So we'll put people randomly into breakout rooms, 10 people each or so, so you can meet old friends again, as well as make new ones. And if you wish to present at one of the next meetings of any information for me to circulate, please do let me know. Very happy to include that information. Everyone is welcome to speak here, and please just get in touch. And so let's get to the program. So we'll have 3 speakers here. The first one will be Stephen Kearnes from Relay Therapeutics, or formerly Google, now Relay, on model validation. Afterwards, we will have Andreas Tosstorff from Roche, together with the CCDC on augmenting the drug design process with experimental protein-ligand interaction information. Afterwards, there will be a talk on DataFAIRy, so how to annotate bioassay data. And I would like to hand over to Steve Kearnes, please, for his talk on model validation. And I was very impressed when I found his papers on the internet. A very important quote is, model validation is always process validation. But I think Steve will come to that later as well. Steve, I will stop sharing and you can share your screen, please. Then.
Great, thanks so much. Let's see.
I think you should be able to share.
How's that?
Looks good.
Okay. Yeah, thanks so much for having me, Andreas. And you know, I really appreciate a lot of the things that you've written in this area as well. And I'm excited to be here. This is a fun group that I haven't had a lot of chance to interact with because I spent most of the last 10 years on the West Coast in California. And as Andreas mentioned, after about 5 years at Google, helping to lead a team there doing machine learning applications in the drug discovery setting. I recently joined Relay Therapeutics as a director of AI research, and I'll be moving to Boston in a couple of weeks. So it's all very exciting and lots of change going on. And Relay's hiring, so come join my team. We'd love to have you help us really push and drive the AI applications in drug discovery. So today I want to talk about the process of generating data, and you know, a lot of this is going to be fairly abstract and also something that I think conceptually that many of you will be familiar with, but I want to highlight some aspects of this procedure and process that I don't think we always think about. My goal is to talk for about 20 minutes to go through some of these ideas and then talk a little bit about some of the work we've done applying these ideas, particularly around machine learning for DNA encoded library selections, and then leave plenty of time for questions and conversation, because I think it's important that we all get on the same page in terms of the nuances around generating data in this field. So you're all familiar with this process. I've seen it represented many different ways. Sometimes it's abbreviated as design, make, test. I like to split it out a little bit broader for applications in early drug discovery, where we include things like compound selection and synthesis. And none of this should be surprising, but I think we all know that the main bottleneck is here in terms of actually making and acquiring compounds and then testing them in terms of the time lag. And there's a lot of really cool work going on in this area, including some work I'm involved in with an effort called the Open Reaction Database to gather chemical reaction data in a structured way to enable machine learning applications to accelerate this effort. And I have some links from that at the end. But today I want to focus on this circle as being a process, being a closed loop. A goal that we talk a lot about in this field is being able to run this process over many iterations, and to do so in such a way that it's an active learning paradigm where we over time gradually gather the data that we need to make our models better, and then eventually come up with compounds that have the best multi-objective properties in this iterative pathway. And this is exactly the sort of thing that happens in drug discovery companies all over the world. But the main differentiator is the amount of automation and the amount of process level thinking that is happening as this cycle is iterated. So let's take a look at just a very generic data generation process. All I mean by this is that there's some black box, and labeled compounds go in, and then selected compounds come out that you then acquire labels for. This is one loop of the process. And the important thing here is that we can write down exactly this sort of information that was going in, and we can write down the information that's going out and compute metrics on that to ask whether this process is actually improving the current state of the world. And I think this is, you know, right away we see something where the world at large is not doing a fantastic job in terms of actually recording and being able to write down in a structured way that can track the provenance of what information went into a decision and what information came out, and the steps along the way to make that happen. So if we try to write down some of the decision points that are made here, this is just a summary of the many things that you would need to write down if you were just doing a sort of vanilla, building a machine learning model, making predictions, selecting compounds and testing them. And right away, this explodes into a lot of things that we have to keep track of if we want to be able to reproduce that analysis, for instance, in the future. We have to make sure that we have an accurate accounting of the data and labels that went in. We have to make sure that we've codified the input representation and the choice of the model architecture. There's lots of parameters involved in exactly how you might train that model and validate it. There's the choice of which compounds you're actually going to apply the model to. And that might come from a commercial catalog. It might come from a list that was enumerated by chemists. There are a number of different possibilities here. There's the actual process of choosing which compounds to acquire. or make based on the predictions of this model and possibly some chemistry, some input from chemists. There's the actual synthesis and then the validation. And the point I want to make that Andreas highlighted as well is that if you're doing prospective work, which is what we're doing all the time in drug discovery, the process of validating whether your model is doing well or whether your procedure is giving you the data you want, this is process validation. It is not model validation. The model represents some of these things mostly on the left side here, but there's a whole bunch of stuff that happens after you train the model that we just often don't account for in terms of validating whether the procedure is doing what we want. Let me go into some more detail. So let's take this simple schematic of I've got some training data and I've got a test data set that I want to apply a model to. If I build a property prediction model, this could be a complicated deep learning network. It could be a very simple linear QSAR model and anything in between. And then there's various hyperparameters that go into this, as we discussed previously. The output of this is a set of predictions for the test compound. And then, this is pretty straightforward. This can all be very well automated. This can be coded up and reproduced with very little effort. But then we come to a fork in the road that is a critical part of the overall process. Often what's done is after this model prediction stage, you have a chemist look at the predictions and decide which ones to buy or to make. And the chemist might be filtering out things that she doesn't like, things that she's seen before, functional groups that she's had bad experiences with. She might be pulling in substructure filters and other automated tools. But at the end of the day, you have a human with their own biases and their own experience and past making filtering decisions on what was until that time a totally automated reproducible process. And it's hard to argue that you could have a reproducible chemist because their state is constantly changing. And depending on which chemist you have, their experience is going to be very different. And so this set of selected molecules all of a sudden becomes a very subjective decision on top of what was originally a fairly objective process. Now, on the other side of things, you could go to the other side of the spectrum and have a fully automated selection procedure where you're using, an automated set of filters, some sort of automated selection criteria or clustering. And this would give you a completely objective process. Of course, the downside is that you sort of lose whatever experience and benefit and chemical intuition that the chemist would bring to the table. And my argument here is that when you're trying to decide between these things, we have to get to a world where a fully automated decision process is the norm, because that's the only way to get direct feedback back to our property prediction model. Let's look again at the list of decisions that are being made here. So we've got all of these things that are coming on the modeling side in terms of decisions about what goes in, how you actually build the model. This compound selection part can be very biased if you have humans involved. And that can affect the types of things that happen after the fact. Let's imagine that if you have a chemist look at things and filter out things based on their preferences, it's very difficult to turn that into actionable feedback that can go back and improve the model. And instead, if you have a fully automated process, changes you make over here, you can directly feed them back into the performance of the entire process. I think that the point I want to highlight here, if I'm not being clear, is that when there are subjective decisions involved, they can dilute or mask the effect of changes that you make earlier in the process. So for example, let's imagine that I went and changed the model architecture because I believed that it could produce higher hit rates for some hit finding procedure. If I change the model architecture here and then have a subjective decision process actually selecting the compounds, if the hit rate goes up, it could be because the chemist made better decisions than the last time. Or it could be because I changed the model architecture. I just don't know. There's no way to do the credit assignment back through a subjective process. Whereas if I have an automated procedure that stays constant and I can control for it, changes I make over here will have direct impacts on the output and I can immediately do the credit assignment to know whether I've actually made an improvement in my process or whether I've just introduced some additional subjective noise. So this is a pretty bold statement, and I'm certainly not advocating that we go and fire all of the chemists or anything like this. I'm acutely aware of the relationships that we need to maintain with the chemists in order to make this stuff happen. And so what I'm actually advocating for is, when you work with chemists, you need to try to understand what it is that they're changing in their selection process and try to feed that back to make a more automated selection procedure that can be reproducible. It's a tighter coupling with chemists that I'm advocating rather than a break or a disconnect, because that's the best way to really find out how the models are doing and to really improve them. You have to take the training wheels off the models in a sense and let them make mistakes. Now, with this as kind of a overarching framework of prospective validation, I want to talk for a moment about retrospective validation and how it fits into this procedure. Often what happens, especially in resource-constrained environments and in academia, is that after you have a model that makes predictions, your selected molecules will be drawn from a set of things that have been tested before, because you don't have the resources or time to go and test things prospectively. This applies not just to random cross-validation or things like that, it also applies to temporal cross-validation. If you're doing things like training a model on everything up to 6 months ago and then testing it on the last 6 months, that is a retrospective study. It is limited to the compounds that have been tested before. And the big caveat here is that you don't necessarily understand why these molecules were chosen or what process chose them. If these compounds were selected by chemists or by some automated procedure that you don't understand or appreciate, the set of molecules that you might want to test prospectively could be very different than the set of molecules that you have available to you in a retrospective setting. And here's another takeaway that I'd like to highlight. If you have a procedure, a data generation process, and you're evaluating it retrospectively, if the set of the previously tested molecules doesn't have significant overlap with the prospective set, the things that you would make if you had the resources to do it, retrospective validation just is not going to tell you that much about how the model is going to perform in the future. The reason for this is that the model doesn't have any skin in the game. If the model is not actually being used to select compounds, then you have no idea how it's going to perform in a real world situation when its predictions are going to be used to make decisions about whether to acquire and test compounds. Instead, all you're doing is relying on some other process that was used to make things before. And so one thing that I wrote in my perspective piece in Trends in Chemistry is that retrospective validation really only tells you about how the model is going to perform if it's never used in the actual decision-making process. And so if you are doing retrospective validation, you have to be careful to consider this overlap between the prospective and the retrospective settings and use that as a metric, or at least a signal about how much information you're getting about future performance by doing a retrospective study. So with that as a background, I want to just briefly dive into a case study where we tried to apply some of this thinking in terms of building models and using either automated or automatable procedures to select compounds. And this is work that I did at Google in collaboration with both XChem and Zebi Therapeutics, which was recently acquired by Relay. If you're not familiar with DNA encoded libraries, the basic idea is that you have a very large combinatorial explosion of small molecule compounds that are made by joining building blocks together. You might have 1,000 potential A building blocks that can react with another 1,000 B building blocks and another 1,000 C building blocks. And each small molecule instance that is created by joining these building blocks together has a piece of DNA attached to it that encodes the identity of that small molecule. So that when you incubate this library with a target protein and then wash it off, you can then sequence the resulting binders, the tags on the binders, and then get counts for the individual instances. Now, there's a lot of caveats here and things about noise that you have to deal with in terms of these selections. And one of the very common approaches is to take these DNA sequencing counts and aggregate them over pairs of building blocks into so-called disynthon representations. This is simply a noise reduction technique that can then be used to classify the disynthons as either positives or negatives based on their enrichment in the overall selection. So this approach is routinely used by many companies and has been used by XChem for a long time. And when we started working with them, the first thing we did was gather these disynthon compounds, assign labels to them, and then build a model to try to predict whether a disynthon would be enriched or not. Now, we built a couple types of models here. Our collaborators at XChem built random forest models using the experience that they had in terms of modeling this data. And at Google, we built graph convolutional neural networks, which are deep learning models that operate on a graph representation of the data. And then the neat trick here was that after this model was trained, we were able to apply it to commercially available libraries. The big advantage here is that oftentimes with DNA encoded libraries, you'll find enriched compounds in the library, and then you'll synthesize them off tag and test them experimentally. That synthesis and testing is an expensive and time-consuming process because it usually involves custom synthesis. And by applying this model to large commercially available libraries, we were able to buy compounds for on the order of $100 a compound and get them in a matter of weeks. So this is a much cheaper and a much more rapid way to use the DEL data for hit finding. We applied our techniques to 3 different protein targets: SEH, ER alpha, and c-KIT. The colors here indicate different levels of potency in the biochemical assay. The black bar indicates the threshold for 1 micromolar potency. So everything to the left is better than 1 micromolar. And then each target has 2 different rows, one for the graph convolutional network and one for the random forest. And as you can see, the hit rates are actually quite good. These are fantastic hit rates for any virtual study. And we believe that this is the largest prospective study of this kind that's ever been done, because we bought nearly 2,000 molecules and tested them to really validate how well these methods were working. And in particular, I spent a bunch of time talking about building models and then selecting compounds, and how oftentimes you'll have chemist review that's involved there that introduces bias that makes it hard to know how well the model itself is doing. For this particular application, we did have chemist review, but we were very, very clear with our partners at XChem and that review should be restricted to elimination of molecules with potential instability or reactivity, things that are going to interfere with the assay. They were not looking at compounds and deciding, I like this one, I don't like that one, based on any of these other considerations. In an ideal, perfect world, we would get to a place where this type of filtering could be completely automated. That's not where we were when we did this work. But we feel that we definitely approximated it here, so that this is a prospective study that can also be a reproducible study that can feed back and then make the models better. And one caveat, or one thing that comes up that you'll notice immediately, is that because we weren't asking the chemists to choose things other than to eliminate these reactive or interfering compounds, some of the hits that we got are not the prettiest molecules you've ever seen. And this is a direct consequence of throwing out some of this chemical intuition and other things that the chemists can bring to the table. And that's why it's so important to couple closely with chemists as we're developing these automated selection procedures, so that we can understand why they make the decisions they do, what types of things they would consider that our current models and filtering procedures are not considering. The more we can learn about that process and replicate it in an automated, reproducible way, the better we're going to get at actually doing iterative optimization of our models and really driving the applications of AI into drug discovery. I wanted to leave plenty of time for questions. Of course, I could talk for a lot longer, but I want to pause here and just say that We're hiring. We recently started a new ML discovery group at Relay Therapeutics. Come join my team with Pat Walters and Patrick Riley and Brian Goldman and others. We're building a world-class team here to really drive the adoption of AI in medchem and early drug discovery. I also mentioned that I'm involved in an effort called the Open Reaction Database. I'm working closely with Connor Coley at MIT, and we have a great governing committee and advisory board from lots of different pharma companies and academic institutions that are generating data. We're really excited about being able to drive machine learning applications in this area to reduce the bottlenecks around the design, make, test cycle and cycle times there. So please go check that out, and you'll be hearing more about that soon as we do an official formal launch. So thanks so much.
Great. Thanks a lot, Steve. And there are a couple of questions already. Raj, do you want to go first?
Sure. Thanks for a great talk, Steve. So when you mentioned in the beginning that you want to let the models make mistakes, it sounds like the real challenge is persuading management to understand that the model is going to make mistakes and they are going to have to synthesize and test a lot of stuff that fails or doesn't work out. And this is not new for people who have used QSAR models, but I mean, it does make it much more clear that in the beginning, these workflows won't do better than chemists. So how have you had to address that aspect? And I guess the related question to that is that if you are saying that this workflow will work, have you been able to make quantitative estimates of how many compounds need to be tested and synthesized to be able to say that your workflow is working?
Yeah, those are great questions. Thanks, Raj. So on the first question, from sort of the cultural perspective, yeah, this is definitely an uphill battle. I think you know, and there's a great paper out just this week in F1000 research about avoiding the hype around AI and things like this. But I think that we have to start cultivating a culture where it's okay for the models to be wrong, and we want to work closely with the chemists to make them better, as opposed to AI as a magic bullet that can come in without any context and just sort of make things better, which I think is a common management attitude, perhaps.
Yeah.
When I was at Google, this was easy because Google is built on doing experiments and A/B testing and making things better through iteration, and that's why Google has become what it is, and so that was not a hard sell there. One of the reasons I joined Relay is because I felt that they had developed and cultivated a culture there where this kind of improvement and iterative refinement was the norm. And computation is baked into a lot of what they do there. And so when I was interviewing and talking with the senior leadership there, I was very clear that like my goal was to come and work in parallel with real project teams and take the training wheels off the models. And I knew they were going to fail, but we were going to make them better over time and get to a place that is better than the current state of the world. And they have supported me in that vision and dedicated resources to that, which is one of the reasons I'm so excited to be here. As far as the statistics and simulations about number of compounds needed and things like that, I don't have concrete answers for you there. A lot of this work is still fairly early as we're trying to figure that out, but I think this ties in really closely with a lot of the active learning literature that's happening here that shows that if you're really trying to drive decisions from a place where you can write down the state, you can get provenance information, you can leverage that to vastly reduce the number of compounds that you might need to make or buy compared to what you might think. And so I think that the more automated we get here and the better we do at really driving process level optimization, I think we're going to be surprised with how data efficient we can be relative to what's actually happening on the ground today. But that's not going to happen at the beginning.
Thank you.
Thank you. And Dominic had 2 points as well. Dominic Ryan.
Yes, thank you. Good to see everybody here. Both of my points are somewhat related. Maybe I'll focus on the second one for the question, but the time validation question is always a race against what other project teams are doing. And the classic problem is by the time you have enough data to truly make a big impact on the project, the team has enough data, they've solved the problem. So it's finding that sweet spot where you can get the biggest impact with the least amount of data and do so in a way that really augments the team. So in the first part of it, it's sort of a quasi-competitive evolution. If you look at time validation, can you show that your validation steps generate good molecules, forgetting how you define good in this case, I suppose, more quickly than the project team does? And related to that is your use in the with the DNA-encoded library experiments, where you're comparing or you're fishing for molecules in the literature. Part of the assessment of how well you do there, I think, is dependent on the evolution of those large libraries, because they've been built with the awareness of lots of people going after kinases. So there's going to be a lot of kinase active molecules in them, or receptors, or whatever it might be. So you've got another time dependency in the same setting. You could go fishing for molecules based on a snapshot of the database 5 years ago, if that were possible to do. I don't know if you can. So how do you get around this notion of comparing yourself against the body of knowledge that's been embedded into so much of the rest of the projects around you?
Yeah, that's a fantastic question, and really good points. So I think part of the answer is that this is a major source of drift, I guess. There's real covariate shift going on in terms of the data that's available and the methods that are available and what people know. I think the only way to really measure your performance is to do experiments in parallel, like you mentioned, where you're working in parallel with a project team and doing comparisons there. The key, I think that on a practical level, the right way to do this is to try to impact projects that are ongoing in whatever small ways that you can, but then also have sort of development projects that don't have those kinds of restrictions, where you can focus on building methods and then sort of doing the research that you need and then transferring that over and say, okay, now we have this like production level system that we're going to use when we actually run in parallel. And so it's going to be a constant back and forth between like generating data in order to improve the methods versus generating data to advance the project. And, there's all of a sudden the resources kind of balloon and explode there in terms of what you need to do it right. But I think that's the way to do it, though.
Yeah, thanks. I think we're always fighting the rule of one that somebody brought up many years ago at a Gordon conference. The number of times that the modelers get to be wrong. Yep.
And Brian Goldman, who joined Relay from Vertex recently, he brings this up to us all the time. You get one chance to be wrong, and then the chemists no longer trust you or the models. And I think that's a cultural thing that we have to do whatever we can to change. We have to show that we have to manage expectations and go in and say, I totally expect this to fail, but here's the value proposition. And then the task of the field is to generate data that shows that value proposition is real.
The value proposition is the problem. So if you're managing multiple projects from a research management setting, then you've got fixed resources that you're allocating across projects. And the more you try to pull into a prospective comparative approach, the greater risk that you perceive to be placing on other projects or on the main project. So it's cultural, yes, but it's financial cultural.
Yep, I agree. I guess I totally agree that that's true. I also am aware that for the last several years, anyone who can put machine learning and drug discovery together in the same sentence can get millions of dollars in VC funding. So I think it's becoming less of a financial issue, and it might be a case where the up and coming companies are going to be the ones that really do generate the data to show that it's real.
Okay, I think, great. Thanks a lot, Steve, for your talk. There's still a few comments or questions in the chat. If you have time, feel free to answer those. That would be great, because I think we need to move on in the interest of time. Great. Thanks a lot again, Steve. Yeah.
Thank you.
Thank you. Cheers. And our next speaker, if you don't mind, maybe stop sharing. Our next speaker will be Andreas Tosstorff from CCDC and Roche, who will tell us about using ligand-protein interaction data for drug discovery. Yeah, looks good.
Okay, I hope you can hear me. Thanks a lot for inviting. Today I want to talk about my postdoc project I did at both the CCDC and at Roche. In particular, I want to show you how we use experimental protein-ligand interaction data to augment the drug design process. So Stephen already touched on the design make test cycle, so I don't have to talk about this anymore. But in particular, I want to say that in structure-based design, one of the key properties we focus on is, of course, optimizing the potency. And the faster you get there, the fewer design make test cycles you need, and the faster and the cheaper, hopefully, the discovery of a new candidate molecule will be. Now, when you want to improve the potency, you have various attributes of your molecule you want to improve, which can be, for example, the conformation of your small molecule, its shape complementary to the binding site, or the interactions it makes with the protein. Apart from potency, of course, there are many other properties such as metabolic stability, selectivity, permeability, toxicity, etc. You have to keep in mind when doing all these structure-based optimizations. And so today, my talk will focus on how we can use experimental data on understanding and improving and guiding chemists to optimize the interactions between your small molecule and your protein. So when we speak about optimizing interactions, we always speak about pairwise atomic interactions between the ligand and the protein. And there are two critical aspects to this. One is the strength of the interaction, which, for example, can be measured experimentally, as in this paper where they measured the acceptor strength of different chemical motifs. So you can see, for example, here with these cyclic ethers, you have different acceptor strength. On the top, you have a strong acceptor strength. If you go down on this pK-BHX scale, you have a lower strength. Second aspect to interactions is, of course, the directionality of the interaction. And for example, in the paper by Wilken, they found that halogen bond strength depends on the contact geometry. If you make a halogen bond, the strongest interaction energy is if you follow this green part of the sphere around the oxygen, carbonyl oxygen. Now, these are experimental, and so this information can be determined either experimentally or through QM calculations, for example. But there is also another way to learn about these aspects of interactions, which is through the statistical analysis of structural data. And this is an approach that has been done, of course, previously in many studies. So the approach is generally to analyze the PDB and the CSD and the structural data therein to study certain aspects of molecular recognition and interactions. And this is a very early an example from the 90s, where they studied, for example, the hydrogen bonds in carboxylate groups. And typical problems that arise when you're doing these statistical analyses is on the one hand, the contribution of covalently bound bystander atoms. So by this I mean, when you consider this an interaction and you you have a covalently bound bystander atom, you might falsely also attribute this contact here as an interaction, even though it just occurs because of the covalent bond. Another drawback in this approach may be the lack of an appropriate normalization when you generate your statistics, and the bias in your data set, for example, when you have a recurring scaffold from a specific series, and you count this multiple times in your statistical analysis. To deal with all of these aspects, Robin Taylor developed the ratio of frequencies approach, which is basically a measure for the interaction probability between two atom types. And Bernd Kuhn later applied this also to protein-ligand complexes, and the rationale is simple. The ratio of frequencies is the number of contacts you observe in a database divided by the number of contacts you would expect. And I will tell you later a bit about how this expected contacts are calculated. Let me tell you a bit more about the RF. So we define it both from a protein and the ligand perspective. This then looks like this. So you have two atom types, and this data is for chlorine on the ligand side. On the x-axis, you see the different protein atom types we have defined. And you can see a higher F value means a high preference for this atom type, and a lower F value means a low preference. And you can see that you have from the ligand and the protein perspective different preferences for such an interaction between chlorine and either of these protein atom types. Importantly, to sum up what the RF value tells you, it informs you about the ligand atom type's preference for protein atom types and vice versa. And these, as you can see in this plot, must not necessarily be the same. To tell you a bit more in detail about what the ratio of frequencies is. So as mentioned before, we have the observed contacts divided by the expected number of contacts. And the way we calculate the observed contacts is by only counting line of sight contacts that occur in our database. So by line of sight, I mean contacts that are not intercepted by a third bystander atom. So for example, this central atom here is in line of sight with this atom here, but it is not in line of sight with this covalently bound atom because the line is intersected by this atom here. And so by only accounting for line of sight contacts, we reduce greatly the effect of this of bystander atoms in our statistics. And to normalize our data appropriately, we have a surface area based normalization term, which accounts for our expected number of contacts. To put it simply, the larger the surface area of a protein atom type in our binding site is, the higher would also be the expected number of contacts. And by dividing now the observed contacts by dividing the observed contacts by the expected number of contacts, we come up with the ratio of frequencies, which is higher. If it's higher than 1, indicates a favorable interaction, and if it's lower than 1, it can indicate for simply an interaction that does not occur as often as one would expect based on the surface area. Additionally, I mentioned earlier that not only interaction strength, but also the directionality is crucial to evaluate and characterize interactions. So we also constrain our RF calculations by geometric constraints. And you can see in this heat map now, again, a plot of RF values for chlorine versus different protein atom types, but now constrained by a contact angle. And just without going through the details, you can see that for this area here, we have a blue bin indicating a higher F value, which represents the halogen bond, which occurs at the extension of the sigma bond along the chlorine carbon bond. And these are known to be favorable and are nicely reflected by our geometrically constrained RF values. And I will tell you a bit more about these halogen bonds in a few slides. Importantly, when we generate such data, we have to take into account to use only high quality structures, to not have any artifacts in our statistics. So we only take into account structures which have a real space correlation coefficient above 0.8, high ligand occupancy equal to 1, and resolution below 2.5 angstrom. And why in particular the consideration of real space correlation coefficient is important is depicted here. So you have two high resolution structures, but only one of the ligands is actually experimentally defined, as you can see by the lack of electron density on the left side, whereas on the right hand side you have electron density and experimental evidence that the ligand is actually positioned in this region. So in our statistics, we would have determined context from this compound, but we would have included this binding site. And an additional aspect we do to eliminate bias is to filter out recurring scaffolds in the data sets, because you can imagine from drug discovery projects, you may have series with recurring scaffolds and only slightly different decorations, and to not excessively shift our statistics by these scaffolds, we eliminate these. And we also do not consider cofactors because their binding sites are typically highly conserved. So again, the interpretation of the RF values, if it's above 1, the interaction occurs more frequently than expected. If it's below 1, it occurs less frequently. And very importantly, if it's non-competitive, meaning below 1, this does not necessarily mean that it's repulsive or unfavorable, but it only means that an interaction occurs, that there are other interactions that are more likely to occur. So back to the halogen bond, for those that are not familiar with it. So when you have chlorine bound to a carbon atom, you have this positive partial charge along the extension of the sigma bond, and this leads to particular interaction phenomena. However, with the analysis, with the ratio frequency analysis, we can see that not all halogen bonds are the same, and in general, the interaction between chlorine and the carbonyl oxygen is highly geometry dependent. So you can see here when this sigma hole does point away from the carbonyl, we have a very low RF value indicating the repulsion between the negative partial charge on the chlorine and the carbonyl, and also desolvation effects. Now, when we have the chlorine pointing towards the carbonyl oxygen, we see an increase in RF total, but we still do not compensate for the desolvation. So the RF value is still below 1. And only when we move the halogen bond out of the plane where the hydrogen bonds are made, we can see an RF total value way above 1. And this correlates nicely with the QM calculations I showed you before, where you can see where this green with this green ring around the carbonyl having the strongest interaction energy for the chlorine halogen bond with the carbon. Okay, so here's another example regarding the halogen bond, and this also relates to the previous slide. So in this cathepsin L S3 pocket, we have a series of different substituents in this para position here on this molecule, and you can see that changing the hydrogen to a chlorine leads to a strong increase in affinity, whereas replacing it again with a fluorine leads to a decrease, and bromine and iodine each increase the binding affinity even further. And when you look at now at the RF values for this binding site, you can see how this correlates nicely. And so for the fluorine, we have a very low RF value. We only have one interaction to this carboxylate group here, which is of course unfavorable because of desolvation and electrostatic aspects. Now, when we insert the chlorine, you can see that it makes the halogen bond, but it is in plane of the carbonyl group. And as we know, as we've seen before, this is not the strongest interaction, but it makes additional hydrophobic contacts which have RF values above 1, and these might contribute also strongly to this favorable binding affinity. And now when you replace the chlorine with the larger bromine and iodine, which also have a stronger sigma hole, you can see that the RF value for this halogen bond increase strongly above 1, also correlating nicely with the experimental data. Here's another study of molecular recognition aspects, similarly to the halogens. Also, sulfur does have this sigma hole at the extension of the sigma bond, and you can see that thiazoles, for example, when placed on top of the carbonyl, also exhibit this this RF value above 1. And we also see for a stacking geometry RF values. For a face-to-face stacking geometry, we also see RF values above 1. Whereas if you move in plane of the amide and H, you have, of course, desolvation effects, which then lead to this very non-competitive interaction. So you can see that RF values are useful to query various aspects of molecular recognition. Here's another example for pyridine, where we looked at the stacking. And often in drug design projects, you might wonder if you have an aromatic ring above an amide, where do you want to put the nitrogen? And we can now query this with the RF values, and we found that the best place for the best atom to put above the amide carbon is any of the pyridine carbon atoms, whereas you want to avoid at all costs having the nitrogen above the carbonyl, as indicated by this low RF value here. And this is, here's an example of such a geometry where you have carbon above carbon, actually in one of the SARS-CoV-2 protease structures from the Fragalysis project. Okay, now that we have developed these RF values, calculated these RF values for all types of ligand atoms, when you are now in a drug design project and you wonder what's the best atom type to put at a certain position to make a contact to any protein atom type, you can now query this and search for this. So if you wonder, for example, what to put above a carbonyl carbon atom, we search for ligand atom types that have the highest RF values here, and fluorine atoms appear to be extremely favorable when put on top of a carbonyl. on top of a carbonyl carbon, so are aromatic unpolarized carbons, and also the oxygens from a sulfonyl group. And similarly, we ran such a search for the sulfur of a methionine, and we find that the sulfur does this type of stacking interaction with amide groups, and is particularly favorable for interactions with the nitrogen from an amide or an urea in this case, but also with other hydrophobic polarized or unpolarized or weakly polarized carbon atoms. Okay, so last slide regarding molecular recognition. We ran a study particularly focused on what interactions to avoid, and on the other hand, identifying these as design opportunities. So we looked at a number of contacts which produce very low RF values, and many of them can be rationalized by desolvation or electrostatic repulsion. There are some interesting cases, such as fluorines on top of the phenyl ring or phenylalanine. In general, halogens should avoid any charged protein atom types, mostly because of desolvation, obviously. And just as the fluorine, the nitrile also should not be placed above a phenylalanine ring. And I can also refer you to our publication on non-competitive interactions here. Nicely, these RF value correlate well with known motifs, such as the kinase hinge binding. You can see when we look at these polarized CH atoms, for example, they all give very high RF values, and this provides useful feedback when designing compounds. We also have interaction typing, which can be useful to interpret the interactions that are visible. And similarly, in the cathode scene, the SO2, which is an important motif for binding affinity, is also reflected by high RF values. Okay, so the talk is called Augmenting, and this slide is really about the augmentation of the design process. And this video already shows you the tool we developed in collaboration with Richard Bartel from CCG, which is highly interactive and depicts the quality of the interactions in your protein-ligand complex, and is updated in real time by whatever design change you make. And I think this had quite a nice impact in the project teams and in design meetings, as a lot of the design ideas in the projects are generated interactively in design sessions with various people, and you can immediately model an idea and get feedback on the quality of the interaction. So you can see when the user builds in these fluorines in this DDR1 example, you immediately get feedback that these fluorines are favorable, indicated by all the blue lines that pop up. And this is actually also reflected in an affinity gain in this particular example. Just some details. We distinguish between statistically significant and non-significant interactions by these thick and thin lines, and we also condensate the information given by the multiple interactions in these spheres. So if you have a large blue sphere, this indicates that your atom will make multiple favorable interactions. Just another application we made, this is just a small example to rescore docking poses. We looked at the enrichment of the .e data set by simply adding an RF-based term to the ChemPLP fitness function. With this empirically identified weight, and we found that by penalizing low RF values and rewarding higher F values, we see a nice increase in the enrichment in 7 out of 8 examples. And yeah, we are currently investing the use of RF values for rescoring even further. So to sum up, we use this RF data derived from experimental interaction data to study and learn about molecular recognition. We visualize it interactively in Mo and use it in project team meetings to assess design ideas. Yeah, we are also investigating other tools to come up with new design ideas, for example, by searching databases for high RF interactions. And we are also looking at rescoring protein-ligand complexes. Yeah, with this, I come to an end. Thanks a lot to especially my supervisors, Bernd Kuhn from Roche, Jason and Robin from the CCDC, and Richard Bartel from the CCG for developing the visualization tool, which had really a great impact in the discovery project. And yeah, thanks also to all the other folks at Roche and CCDC. And with this, I'm coming to an end. Thanks again for inviting.
Great, thanks a lot, Andreas. I think there are a few, at least technical questions as well. Anirudh, I think, asked the first question. What is meant by appropriate normalization? Anirudh, do you want to expand on that? Or?
Yeah, sure. Appropriate normalization, if you would simply count interactions without normalizing them, you would count interactions more frequently if a certain atom type occurs more often. So you have to normalize by the occurrence of the atom type. Otherwise, if you have a lot of carbonyl oxygens in the binding sites more than other atom types, you would you would get higher values for these, of course.
Yeah, the other one, that's a brief question. I will just read that as well. Are the RF values actually affected by the export chemistry in the databases? And I think the question is about biases, for example. Yeah. Can you expand on that as well?
So we looked at this, and as I mentioned, this is why we remove cofactors and recurring scaffolds. There might be biases to some extent, but yeah, we cannot exclude it completely. But it, I think from the examples we've seen, in general, the values are reasonable and go in hand with chemical intuition and other approaches and methods. Yeah.
Okay, great. Yeah, thanks a lot. I think there's at least one more question, and maybe you can answer that in writing, Andreas. Yeah.
Sure.
Great. Thanks a lot for your contribution here.
Thanks to you.
Thank you.
I'll stop sharing.
Thank you. Yeah, and we will come to our final contribution for today. And so that will be about DataFAIRy. So the question of how can we annotate ontologies, bioassay data, which is important if you want to do large-scale mining or model generation. And this is what the Pistoia Alliance will comment on. And that's Nick Lynch, as well as Isabella Feierberg and Samantha Jeschonek. And I think Nick is presenting, right? If I'm not mistaken.
I think Isabella's got the master slide.
Okay. Very good. Yeah, just make
Isabella,
that.
I think you're on mute.
You're still on mute, Isabella. Still on mute, I think.
Yeah, let's see. I asked her to unmute here as well. Maybe that works. Or...
Unmute.
Yeah, no, it's fine.
Great, thank you.
No,
Sorry about that. I couldn't couldn't get to the unmute button through the app. So can you see my notes page, or can you see the full screen?
We can see the slide. That looks
The
fine.
slide itself. That's great. Okay, so thank you so much, Andreas, for inviting Samantha, Nick, and myself today to this meeting and to present the DataFAIRy bioassays pilot project. So just to introduce ourselves, I'm a computational chemist at AstraZeneca. I've been with AZ for 17 plus years. Samantha Jeschonek is she's a product manager at CDD for their semantic suite, Bioharmony. And Nick Lynch, he is the founder of Curlew Research, but was also a founder of the Pistoia Alliance. And we're here today to talk about the DataFAIRy project, which is a pre-competitive multi-pharma collaboration about enabling data science. And as you can see on the bottom of this introduction slide, the companies that were involved, apart from AstraZeneca, are Novartis, Roche, and BMS. And we were also collaborating with the collaborative drug discovery for the tech part. There are many aspects to this project, and what I wanted to focus on today is the cheminformatics aspect. So we'll start with an example. So say you are a cheminformatician at a pharma company and you want to build a model that predicts the kinase selectivity profile of a compound. So what do? You get all the kinase activity and selectivity data that you can from your in-house databases. You can take them from the public domain or from your collaborators. And as we heard in Stephen's talk, one very important aspect of successful modeling is the training data that you get. And this is exactly what this talk focuses on, the training data and the labels on that training data, which we call metadata in this project. You go ahead, you build your model, you use your model, you evaluate your model, etc. You test compounds, and when you have new results and new compounds, you update your model with new data. And to be able to be successful with this, we really want to have the best data sets possible to build those models. So what we really would want is high quality data, data sets that we know are taking care of any bias problems. They should be well annotated, and there should be a lot of data. So how do we get a lot of data? Well, basically, you could envision merging your data with your partner's data and with the public domain data. But the problem is that it's a little bit tricky to merge these because those data sets are often siloed and described and labeled in very different ways, and maybe they also lack metadata for you to be able to take the decision. For instance, for kinase, if you're building a model to predict allosteric inhibition of a kinase, the ATP concentration is interesting for you. And often that is not available in the public domain. Maybe in your in-house database it's there, but you really want to know that you can merge those data sets together to be successful in your model. Right. So if you are able to do that, you would be able to merge all those data sets into one. And that is really where you want to get in the end. So we can conclude here that there is a need for fair public domain data. Now, FAIR is an acronym. It means that the data should be findable, accessible, interoperable, and reusable for data scientists. And you want those labels and the annotations to be of high quality, and you would really want to use a common data model and public ontologies so that you know that what you call a target in your internal database, your partner's definition of a target is the same. And whatever you find in Chembl, that is also the same. There is a lot of data in Chembl and PubChem and other databases out there, BindingDB. And that's great, but often we don't have enough metadata, enough detail in that metadata to answer the questions that we would like with data science today and in the future. At the same time, there's a lot of investment going on, like we heard in Steve's talk, in the machine learning and AI sphere, and also at making data fair within companies. There are also plenty of pre-competitive collaboration about how to make data fair. So toolkits, etc. And some of them are driven by the Pistoia Alliance as well. Now, for you to be able to merge all those data sets together, you would really need to curate whatever is available in the public domain. Now, that is an expensive exercise if you do that to the level of detail that you would really need. But it's being done to some extent. Companies pay money, and Campbell pays money also. Other databases pay money to third parties to curate data sets and papers and patents and other things. So the idea here was, if all companies or all big pharmas, for instance, want the same public domain databases to be fair and to be curated, they all want a common data model. They all want high quality data, and they all want to use them in more or less the same ways to use for modeling and for ingestion in their internal knowledge graphs, and so that their scientists can inform themselves about experiments that have been published, etc. Why not just share the cost of doing that? And so this DataFAIRy operational model was proposed in 2018 within the Pistoia Alliance. And the idea here, in the middle, you see the square, it says DataFAIRy partners. That would be the partnering pharma companies, for instance. It could be other types of organizations as well, like CROs or publishers, but let's just focus on pharma companies in this case. So let's say a bunch of pharma companies come together in this, and they say, okay, we want this database to be verified. We want to curate X number of experiments from papers, and we pay for it once. And the idea here is that the partners would select what data sources to make fair and to curate. And then we would have experts, domain expert and data curation expert, to do the actual curation work. Bring it back in fair format for the partners to ingest into their internal systems. But after some grace period, let's say 18 months or so, just upload that into back into the public domain, so that other people in the scientific community and in academic labs, etc., across the world can use that metadata to their benefit. Because it originates in the public domain, there is nothing proprietary about it. When it comes to internal data within the pharma companies, that's a different story. Here we're only talking about public databases that everybody has access to anyway. And so we wanted to run a pilot case for this operational model. And we thought small molecule bioassays is a good pilot case because there are many different people that can use this type of data. For instance, if you're a biologist, you're going to set up an assay. If you can go into a database and say, hey, for my receptor that I haven't tested yet within my company, what's out there? What are the different assay setups? What's available in the public domain? Or once you're actually going on, what are the tool compounds I can use? What are the assay conditions? If we can get to that level of detail, that would be just amazing. Another use case is, of course, chemogenomic model building, like I described in the first slide, where a computational chemist or a cheminformatician or a data scientist goes in and wants to develop a model. And so we formed a project team within the Pistoia Alliance, and these are the team members. We have 2 people from Novartis, people from BMS, Roche, AstraZeneca, and we partnered with CDD and also with the Pistoia Alliance that were the project management part. And I think I'm going to leave this presentation now for Samantha to continue.
Thanks, Isabella. So when we started the pilot project, we really wanted to be a feasibility study for a much larger initiative. We first need to see, does this sort of public curation work? Can we get the data back into the public sphere? And does it answer the business questions, or is it useful as we move through the project? And throughout it all, we wanted to, of course, capture our learnings, and share those with the public as well. So we decided to curate about 500 public domain assays, convert them to a FAIR format using a data model that the stakeholder team and CDD helped design, and then upload that data back to PubChem. I think you can go to the next slide, Isabella. Perfect. So FAIR is such an important topic nowadays, and I think it's always important to take a step back and remember that we're not verifying things just so we can say that it's fair, just for the sake of being fair. There should be a purpose or an intent behind it. It's usually because you want to be able to use that data. So it was really important when we started this project to have a defined set of questions or business questions that we would want to be able to answer if we had these assays verified. And we tried to set out with a set of business questions that were covered a number of different areas. You can see the 5 sort of categories below, things like biology-oriented literature mining for discovery planning. And those questions might be like, for my new inhibition project, I want to find all papers describing functional assays for measuring my target inhibition that are reported gene assays, but don't actually use luciferase kits. We wanted to be able to answer assay technology-oriented questions. So how have artifact assays for ADP Globe been set up in the public domain? What are their differences? We wanted to look at chemistry and tool compound-oriented questions like Isabella mentioned before. Are there tool compounds out there in the published literature? How is it measured? We wanted to look at specific assay questions. So give me all the in vivo clearance data that was run in canines. And finally, as Isabella mentioned, we wanted to look at chemogenetic modeling data. So building a training set for virtual screening for a particular target. The next slide, Isabella. So to proceed with this pilot, we needed to find a technology that would help verify these assays. And to do so, the team put out an RFP requesting multiple vendors to send in their particular proposals. Specifically, we were needing a tool that ensured really high quality, accurate metadata capture with the ability of human intervention. And so looking through those RFPs, the tool that sort of met all the boxes was CDD's bioharmony annotator, which you may previously know as Bioassay Express. And this tool works with a patented hybrid machine-human learning approach. What we did was our team came up with a agreed upon template that's linked to public ontologies. And then what the tool itself does, it uses a combination of natural language processing, machine learning, correlation models to predict the annotations for a particular assay. A human then can go in and say, that annotation is correct or that annotation is incorrect. And then it has a dynamic learning process behind it that as you annotate more, as you submit more suggestions, the machine becomes smarter. This kind of iterative review was really important because it both sped the curation process and allowed us to ensure that the metadata that we were producing was highly accurate. One of the tools that is included in this bioharmony annotator is something called OntoloBridge. And that's really nice because it allows you to even submit provisional terms as you're annotating an assay. So our scientists were never stuck with the situation where there wasn't quite the right term and the ontology selected in order to annotate the assay. We could just submit a suggested new annotation term. And in fact, we submitted over 100 to the bioassay ontology that came out of this project, which I think is really, really interesting. So Isabella, I think you can go to the next slide. So in terms of the assays selected for this project, we wanted to choose assays that were diverse in nature and diverse in their format as well. So about 42 of the assays we selected were from PubChem and NCATS assays, and those assays were described as free text on the PubChem page. The next set of assays, a large bulk of them, were a commercial panel. They were the Thermo Fisher Z-Lite kinase selectivity assay. Those assays were formatted in a structured table format embedded inside a PDF. And then finally, our last group of assays were those assays that had data deposited in Chembl. The target for the assays was EGFR, but the assays were described exclusively in sort of the open access publications. So PDF, supplemental material, different citations. So of the 500 assays that went through the hybrid human machine curation process, 100 of those were then selected to an additional rounds of manual review, again, to ensure the accuracy of the metadata. So 100 of the assays is about 20%, and a lot of the learnings that we took are really from the type, they're biased towards the types of assays we decided to quality control or QC. The largest portion of assays that we looked at were these Chembl assays, the ones that were described in the public domain literature. And those actually proved to be the most difficult. And that is because there is actually information about the assays missing in the source publication themselves, resulting in our curators having to citation jump from one reference to another to look at supplementary materials and to do their own literature searches. So many of those papers went through additional rounds of QC, I think up to 4 or 5 rounds of quality controls, what we needed to really have everybody dig through the literature to find the right answers. Next slide. So looking at those 100 manually curated and reviewed assays, we wanted to evaluate how well they were annotated, how well they answered our business questions. And what we found was there was a consistency in the amounts and types of information that was excluded from the public literature. Often that was centered around controls and different concentrations. What this means for the project is that your business questions really dictate whether your annotation is a success. You might be running a project where not knowing the concentration of your inhibitor compound it's not critical. And so in that case, it's okay the information isn't really provided in that source literature for your purposes. For somebody else, it might be different. But perhaps you're running a project where it's crucially important that you know the amount of, say, ATP used. Now, if you had looked at the literature sources that didn't contain that information in the source material and time was required to jump between sources and eventually not even find that data, you would be in a very difficult spot. So it would take a lot of effort to try and get that information out. So what really drives the types of papers that you should select and the type of assays that you should look at and the format or the data model you should start with are those business questions. Next slide, Isabella. So we came out of this project with a number of learnings that we'd like to now share with the group. And the first is, as I've been hinting at, if the data does not exist in the source material, if you have to jump through citations, if you have to look at supplements, that's going to increase the cost and the time and resources put into your assays. You need to choose wisely the type of assays and the material that's provided in those assays that will answer your business questions. What we also found is that commercial assay panels were really the easiest to annotate. They're low-hanging fruit, and they're really nice because they are widely used. Annotating an assay panel, the general overview of that assay once can actually apply to all the targets of that panel. So you can put the time in once, change a few terms, and suddenly you have 200 plus assays annotated. But the problem we found with those is that the literature sometimes doesn't include details about those assay panels. They will often reference a panel, and it's fine if that panel is then still active and exists, and you can look for yourself to find the necessary assay conditions. But often we found assays, panels that were no longer in existence, or they changed name and changed company, and we couldn't actually find out how these assays were performed. And that becomes problematic, again, because one assay panel is actually 200 different targets, and you start propagating those errors between papers that reference each other, referencing a panel that no longer has data or information metadata about it anywhere. So that really leads into the last 2 points there. It's really important to capture this information at the time of submission or at the source. And wouldn't it be just absolutely wonderful if we had these published assay protocols around not only commercial panels, but other assays that were in a standardized format, a standard data model, and in a public database. It would be able to answer your business questions. You'd be able to find that data quicker and faster. And I think as a whole, the scientific community would greatly benefit. The final learning we came from this is really around the tool itself, which is that a fully automated system isn't fully accurate. And when you're really going for highly accurate metadata for your models, it's important to choose a tool that allows that combination of human and machine learning to speed the curation process. And unique to our pilot project is that we really wanted good work practices. We worked on audit trails and versioning and iterative quality control. And that's something that I think benefited the quality of the data we were able to output from this. It's going to be uploaded to PubChem, and is also a series of steps that we intend to include in any additional phases of this project that come out. Is there a next slide? Isabella, I can't quite hear you.
There we go. So, you know, is this really useful is a question you could ask yourself. What's the value? Can we prove that we get a return on investment? These are questions that we also discussed in the first talk during this meeting, right? In Stephen's talk. Actually, within AstraZeneca, we already have a use case. So one of my colleagues, Lewis Mervyn, who is a machine learning expert, actually used the output from the DataFAIRy pilot. So happy to share those details that he took them to a pre-competitive collaboration that he is part of and used it for chemogenomic modeling. So what's going to happen now? Well, we would like to continue this, and we would like to scale it up, and we would like to bring in more partners because it will be more return on investment as well. We would like to optimize the process and the data sources. Like Samantha mentioned, it's really good to choose your assays wisely or your papers wisely. We believe there is a way we can do that. And we want to also streamline the tools more and also make sure that we keep the quality control, because quality is key here. If we don't want quality on our detailed metadata, we could do something else. And we would also like to work on quality metrics to actually be able to assess the quality. And the other leg, so one leg is to scale up the number of assays and papers that we verify. The other leg is that we would like to define and promote a community standard for assay reporting, similar to the PDB for crystal structure. Like we saw in Andreas's talk before here, the PDB is really well annotated. If we could do a PDB for assays, wouldn't that be just amazing? When things would mean the same to everybody publishing an assay. And we will also be aligning with vendors, so assay vendors, basically those commercial companies that sell, for instance, selectivity panels, as well as publishers of papers and government agencies, because there are overlapping interests from all of those different parties. And we would also, of course, want to attract new project members, which will enable sufficient funding to start the next phase. Now, a lot of people were involved in discussing this idea before it was even put into the Pistoia database of ideas. And I'd like to just say thanks to all of these people. And then in the end, I have a slide on why bother? Why would your organization want to join this? Well, it brings a lot of value to the scientific community to make the data more fair. It will increase the body of fair data. There will be a PDB for assays, which is great. That would be accepted data standard for assay metadata. It will be cheaper for everyone to access that fair data. And as a long-term goal, we have also posted that we would potentially be able to lower the cost of regulatory submissions. Yep, that was my last slide. I'd like to open up for questions.
Excellent. Thanks a lot, Isabella, Nick, and Samantha. So the companies should just get in touch with you, basically, yeah, in case they want to join that effort, yeah?
Yep.
Yeah, okay, no, sounds good. Okay, and I think Chris Sutton had a couple of comments. Chris, do you want to go ahead?
I mean, I can see, it's Nick here. I can see some of Chris's questions in the chat. I can probably try and we can obviously do this a bit offline too, and maybe publish some of the answers. But Chris asks about the difference between chemical assay annotation, PubChem, and this. I think, as both Isabella and Samantha said, it's about the level of detail and what you're using it for. And I think what was attempted to do with the sort of minimum information set that was developed for this project, it's trying to get something which is maybe an enriched set that has existed before.
Hi, hello, Nick.
Hello, Chris.
Oh, hi. Hi, Isabella. Nice to hear you. Great.
Hi, Chris. How are you?
Yeah, just quickly. Yeah, of course. The trouble is, as Kemble goes into PubChem, you get slight, and you look at the CDD bio version, they're all slightly different. Okay, so you've got a non-standardization thing there already. Okay, so I think the only things I can carry on with is that we've all said for years that the publishers should ensure assay standardization. I don't know how much hope we've got now, and Strender has been around for donkey's years. That's an assay standard, but it hadn't had any take up. I guess the other thing is we see now, in fact, the British Journal of Pharmacology, that protocols.io are positioning themselves as an assay repository. Are they really going to be up to the task?
So I'll comment generally and let Isabella get maybe more specific. But one of the things we're collecting here is not just step-by-step instructions about how an assay was performed, which is really what protocols.io focuses on. We're really looking for those higher level categorization of an assay that can let you know, can certain assays be aggregated together for different data models.
Assay ontology.
Yeah, using assay ontology, exactly. Yeah. So that's one key difference. In terms of, and I answered this in the chat a bit, the non-standardization of, say, Kemble versus PubChem versus annotator, we actually brought in the metadata fields that existed in PubChem that if they had been annotated, they had been marked up into our data model for DataFAIRy. So we are transferring those learnings and information and not just kind of replacing it with our own. The nice part about the certain fields is that you can link the field, say assay type itself, to an ontology. So it's not just a standalone term. And you can annotate it with an ontology term that is also linked to the public, right? So everything you're keeping kind of interoperable and consistent. I'll pass it to Isabella to add a bit more information or flavor to that.
Sure, thanks. So Chris, yeah, so people have been talking about this for ages, right? Like you mentioned, and tried to make it happen. And there was BARD and there was this and there was that. Now we think it's the right timing because of the investments in fair data and in AI and machine learning. We think there is enough momentum to get this.
You can tell the journals they're not fair unless they do it.
Right.
Quite how much traction that's going to have open to doubt, but anyway, as it is a thing. Sorry, one last point. When I talk about fair, I often add the extra R, reproducible. Now, I don't. Has the project kind of even tangentially considered this issue?
The reproducibility of an assay.
Yes.
Well, Samantha is the biologist here, but I I can try to address this on the level that I can, and then I'll leave the mic to Samantha. So reproducibility, for that to happen, you really need to be very, very detailed, right? You need to have control over a lot of stuff. As we saw in those assays that we annotated for EGFR from Kemble, those would not have been able to be reproducible because we didn't have enough data there. So even those that were published, and were referencing a commercial assay panel that had been available 20 years ago and did not contain a persistent link to that description, even though, sorry.
That's fine. No, that's kind of what I expected. I mean, as we all know, the issue going forward with this data, which I think is a fantastic initiative, if you're up with if your spread of published results for the same compound, same target are tenfold apart, you should think what you can do about that issue as well as just having the protocols.
You mean to scale the results or to normalize them?
No. So if as we all know, so nominally same assays, same target, same drug, assays, the distribution of assays being about tenfold, right? Massive. It's tighter in some circumstances, tighter for some sets of GPCRs than other targets, or kinases being, on a good day, reasonably congruent. But as you said, unless they declare the ATP concentration on the screen. So yeah. I'm just saying, I think this is fantastic, but I'd consider the reproducibility in the variant sense as also something you should be, the community should be looking at going forward.
That's a good point, Chris. That could potentially be added into the data model if we have some type of quality metric of the actual results of the assay. Then people could filter on those. They only want stuff that has a low variability, for instance, or they want stuff that has high variability to make their AI model more robust. So that could potentially be discussed. And thank you for that. Samantha, I'll leave the mic to you.
Yeah, really
reiterating what you said. I think. Thanks, Andreas. So time I shut up.
No, but Samantha,
I can actually go down the road and have a coffee with Isabella.
That would be nice.
All right, we're on.
Samantha, go ahead. You still want to comment, I think.
Yeah, so it's a great point, and the data model and the templates that we put together are totally configurable. So if there are suggestions like this, we absolutely welcome them, both in this format, or if you'd like to join on, as I said, on the next partner to the project, right? You can help influence and discuss what these data models are, and we can make sure that it's answering kind of the business questions you're interested in.
Exactly. I mean, I posed the question, but I assume you're encouraging academic groups to join in as well.
I think anybody.
So yeah, okay. Well, first I can recommend, I can prod them the Guide to Pharmacology and Binding DB. Yeah, both of them would be more than willing.
Okay, great. Thanks, Chris. And I think Dominic, he's following up on the previous question. Dominic.
Yes, thanks, Andreas. I wanted to mention or address this question of reproducibility. It's actually a bit of a serious problem, even within a given project team. Maybe not reproducibility so much as the sort of the window of uncertainty of results. If you run a really high quality enzyme assay in-house with great controls, you then look at the SAR that you get. I have trouble believing things that are more than like a twofold difference in values. You start to see more than twofold, you start to believe there's something real going on. Cell-based assay, 5 to 10-fold. If you now start to move that across labs, that grows. If you move that across time and labs and continents, I think it becomes extremely challenging to try and put things on the same level. My hope for this is some kind of relative SAR. That's the best way I find to line up assays to try and attach some sort of relatability to them, and implicitly, therefore, some sort of reliability as well, reproducibility as well. So if you have done anything in that world, I'd love to hear about what you can do in terms of lining up relatable compounds.
Wouldn't it be great to be able to do that, Dominic? Thank you for bringing it up. It is really a challenge. For instance, in a company like AstraZeneca that has multiple labs, this is really important that if you transfer an assay from one lab to another, especially across continents, or people have different setups, and assay variability can happen because somebody has lint on their lab coat. This has been seen. I read something about that, or using a different plastic or a different batch of something. So a lot of little details can really make a big difference. So I think, you could possibly run the same compounds. If you want to reproduce the assay, take a bunch of those compounds, of reference compounds, and run them in your setup, then scale the results of the ones that you're trying to merge it with. I'm not sure there is a free lunch here anywhere, or like a quick
maybe
win.
Not free, but in cases like kinases or sort of standard things that have been around for a long time, there's a fair likelihood that folks will have used some of the same compounds, and that may get around some of the ambiguities that these are often presented with.
Well, that is true, and you could use the same tool compounds, but the other thing is, like, even within your tool compounds, there is variability because they might have a different synthesis protocol. There might be different contaminants. It's always a challenge.
Agreed.
Yeah.
Okay, great. Thanks a lot, Dominic. Oh, Chris, you have a final comment? Because I think afterwards we need to wrap up.
Now, Dominic, well, you could push this back to the journals side again in saying authors should have higher numbers of replicates. That doesn't remove systematic error, as Isabella's just given examples for, but at least some when you get a plus or minus with different stats of an n of 3, that's part of the problem. So we'll see if we can push back to journals for that, and the inclusion of tool compounds in as assay calibrations.
It also comes down to funding, whether it's academic.
Absolutely. Yeah.
Okay, great. Thanks a lot. And I should say, DataFAIRy, similar approach is certainly something we need in order to learn from data. So really appreciate that effort. Yeah. And thanks a lot for your contribution, Isabella, Samantha, and Nick. Much appreciated. Okay, so I would like to thank everyone participating, everyone presenting in particular. And please let me know if you would like to present at one of the next Cheminformatics meetings. Everyone is welcome to present. And please stay on the line as well. So we'll have our pub night. I will assign people to breakout rooms. So there will be 10 people or so per breakout room. So please stay on the line. And yeah, we will be surprised to find your room. Okay, thanks a lot for joining, everyone. See you next time. Thank you.